This is Dr. Vishal Gyawali from Queen's University, Kingston, Canada, and it's my huge pleasure today to discuss about a paper we have recently published in the Lancet Family Open Access Journal eClinical Medicine called a correlation analysis to assess event-free survival as a trial level surrogate for overall survival in early breast cancer. As a background, the FDA has published the table of surrogate endpoints that they had used in the past and they may use in the future for approval of drugs, and which can be accessed through this website link here. And for cancer drugs, they have listed a variety of surrogate endpoints for different tumor types. And in a previous paper, also published in the same journal last year, we looked at what was the evidence behind using the surrogate endpoints in the table, and we took the case example of breast cancer and we looked at the level of validity for each surrogate endpoint there. And this is what we found. We found that for most of the surrogate endpoints listed for breast cancer in the table, the level of surrogacy was either not good enough or it was not even tested. For example, in the case of event-free survival, there was no validation study. So the conclusion in that paper was for event-free survival, there was no published studies available and it's an area of future research. So our current paper is directly a result of that. In this current paper, we try to fill that void by doing a correlation analysis to assess event-free survival as a surrogate for overall survival in early breast cancer. And we came across 8 studies in the neoadjuvant setting which looked at both EFS and OS, but unfortunately Keynote 522 does not have OS data, so we did our correlation analysis on 7 other studies. And we found that actually there is a moderate correlation between the log of hazard ratios for OS and the log of hazard ratio for EFS. But the confidence interval is quite wide and it ranges from negative to positive. And similarly with the coefficient of determination, although it looks moderate strength, 0.76, the confidence interval is quite wide. And this is probably because of the low sample size that we have. So we conclude that although there is a moderate correlation between treatment effects in EFS and treatment effects in OS, but because it is not significant, probably it's not valid for regular approval, but can be used for accelerated approval. Thank you.